cell cycle. They comprise interphase, and then and interphase is G1S, G2 M phase, and the M phase is mitosis. And mitosis, prophase, metaphase, anaphase, telophase. So we'll go through all of those. Now, your interphase is where period between cell division, the chromosomes are visible under the light microscope. This is the time we can see the chromosomes. Now, G1, which as you can see is a massive part of the phase, is where you have metabolically active. These cells duplicate their organelles and cytosolic components, and replication of the chromosomal DNA begins, basically. So everything, it's the gap phase. Gap phase 1, a lot of stuff's happening. In S phase, it's replication of DNA and the centrosomes themselves. If you don't know these words, don't worry, we will define them shortly. G2 is cell growth, enzyme protein synthesis, replication of centrosomes are now complete. So those are 3 different phases. So you've got your organelles, your cytosolic components, chromosomes begin, then replication of DNA and centrosomes. The centrosomes are complete, the cell is growing, enzyme and protein synthesis is occurring en masse. M phase is mitosis, and the M product is that a parent cell produces 2 identical cells with identical chromosomes, the 2M, the diploid, all visible under the microscope. Okay? So in prophase, so this is all mitosis now. So we've got interphase at the top, then we've got prophase, metaphase, anaphase, telophase. So in prophase, the chromosomes condense. The nuclear envelopes, so you can see the thread-like chromatin in interphase on the top right there. The chromosomes condense, envelopes are not apparent, and the centrosomes polarize. In metaphase, the centromeres of those chromatids pair up at the metaphyseal plate, so we have plate formation. Anaphase is apart. So metaphase is middle, pair line up. Anaphase is apart. Those chromatids separate, so they're pulled to opposite poles. In telophase, the nuclear envelope and nuclei reappear. Chromosomes resume their chromatin form, and the spindles disappear. Okay. Now in meiosis, unlike 1 round mitosis, it takes 2 rounds, and we go from a diploid to 4 haploids. So we go from 1 2N to 4 Ns, and they have undergone they have the potential to have undergone homologous recombination, so they're not genetically identical. Okay. So the quick word on the play is these key definitions. Chromatids. So it's a thread-like strand that a chromosome divides into during mitosis. Okay? The chromatin is an umbrella term for protein, DNA, RNA, the constituents of a chromosome and a chromatid. Right? So as the chromatin is a thread-like strands before they're condensed, your chromatin is the content of the chromosome and the chromatid. Okay? The chromosome is condensed chromatin. Homologous pairs recombine in meiosis. You have 23 pairs in each somatic cell that's diploid, okay? MT is microtubule. Tubes say protein for structure. Transport cellular division. They form the mitotic spindles that pull the thing apart. The kinetochore is a protein associated with the centromere, which is that constricted central point of the chromosome. It's the exact point that the mitotic spindle will attach. So a lot of texts will just ignore the kinetochore, and they'll just say the centromere. But the centromere is the point where that chromatin information of the chromosome constrict together. It's the kinetochore protein to which the mitotic spindles, those microtubules, bind. So it is important to differentiate. The spindle is a complex of microtubules that separate those chromosomes to the poles. Okay? The centrosome is an organelle formed of centrioles. It's the mTOC, the microtubule organising centre of the cell. Okay? So centrioles are tubulin polymers that form the mitotic spindle. So the centrioles are almost like the factories that are forming the spindles, okay? The centromere is the region in which the spindle attaches via that kinetochore to the chromosome. So those are quite complex words, worth knowing. How is the somatic cell cycle regulated? So CDK cyclins are very important. So here's an example. So for example, CDK4 and site D will phosphorylate RB, and RB arrests the arrest interphase, so we stop at G1. So to phosphorylate RB, we can progress to S. Then we need those two and those two to progress from S to G2, G2 to M. The take-home message is isomers of CDK and psych are vital for progression throughout the somatic cell cycle. So now let's come to PMAT. This is just a summary of mitosis again. So the chromatin condenses into chromosomes. Okay, so remember we said that the chromatin is the constituents of a chromosome or a chromatid. The centromere holds the pairs together with that kinetochore protein. The tubulins form mitotic spindles, latching to that kinetochore. The microtubules lengthen and the centrosomes are becoming polar. Nucleolus in the nuclear envelope disappears. So this is what's happening. Now, in metaphase, we're forming that plate, right? So the microtubules of the spindle align centromeres of the chromatid pairs at the centre of themselves, and this is the metaphase plate. Okay, so you can see we've got that beautiful alignment there. Microtubules align the centromeres, so we've got those microtubules, boom. Anaphase is apart, right? So centromere splits, dividing each chromatid pair to the poles of the cells. We now have sister chromatids that could also be termed single chromosomes, so we can use them interchangeably now. Boom, they've been pulled apart. Telophase. The chromosomes uncoil and revert to chromatin. So remember, if it's coiled, it's a chromosome. If it's unfurled, it's chromatin. The nuclear envelope forms, the nucleoli reappear, and the spindles break down. And that is PMAT.